At the beginning of class today, you should get an article about nature that has three articles actually in it. I will explain at the end of this video exactly what you are to do with this. So, sorry I couldn't be in class today. Hopefully you enjoy my video. Alright guys, so last class we left off with you guys did a web quest over the different scientists who are responsible for determining the structure of DNA and what they kind of got to. And we got up basically to Watson and Crick. I will collect that web quest next class, so don't worry about it. On Monday I'll go ahead and collect that. So just keep it for now. But basically all the different parts of these images here you can see had to be kind of responsible for different scientists. And like I said, you are going to be responsible for that on the test. What we're going to focus in on is what did Watson and Crick do, and basically what were they able to determine. Watson and Crick built a model of double helix, and they used X-ray, the data from the X-ray crystallography, along with chemistry of the DNA, understanding how it fit. Most of the work, once again, was not actually them doing the work. It was taking other people's work and applying it to the problem. They were very good at that. They were very good at looking at the problem, seeing other people's data, and then actually using it. One of the big people, obviously, they got their data from was Rosalind Franklin. And Franklin concluded that there were two anti-parallel sugar phosphate backbones. Now, she gets this after a long process, and basically what that means is, the DNA strands are going to run in opposite directions. Watson and Crick originally believed that they ran in the same direction. Okay, and so in other words, A would have had to bond with A, and T would have bonded with T. Okay, and so they did have the fact that the nitrogen bases paired with each other, and they knew that they were in the interior, but they did not get the bonding correctly. Okay, so and when you do that, what ends up happening is you don't get this uniformed width, because if you think about purines and pyrimidines, a pyrimidine is a single ring, where a purine is a double ring. When you bond those two together, if you bonded two pyrimidines together, and then kind of like this, you notice the shape of the DNA would not be parallel. It would kind of bow back and forth. So the only way that they kind of could figure that could work is that the purines had to go with the pyrimidines. Now, how I always remember that, the word pyrimidine is obviously the bigger the word, but it's a smaller molecule. It is only a single ring. Purine is smaller. It is the double ring. And you can see exactly what I'm talking about here. Notice it would not create this parallel pattern if we had the bonding like so. So the way we had to do it, the best way is a purine with a pyrimidine. Purine is the double, pyrimidine is the single. And Watson and Crick realized this, but not only did they realize this, along with the data from Franklin, but they had to determine that there was a specific pairing that they had to associate with. They knew that it wasn't just going to be a random assortment, that there had to be some rationale for it. And when they looked at the actual structure of the different letters, one of the things they realized was, and they got help from another man in their office, was that the type of nucleotide that they were looking at, what state it was found, was not actually what was found in the DNA structure. And when they reevaluated it, they noticed that there was a natural association that adenine would only bond with thymine and guanine to cytosine, A with T, C with G, which both of this data confers with previous research that was done by Chargaff. And Chargaff ratio basically said that A equals T, G goes with C. Now, Chargaff was a really good scientist and realized he didn't want to jump to conclusion, so it didn't mean that they necessarily bonded together. It was kind of funny. There's an interaction between Watson, Crick, and Chargaff, and Watson says, "Oh, A must bond with T and G must bond with C." This is early on in their research. Chargaff says, basically very matter of factly, no, it doesn't have to work. All it means is A equals T, G equals C. Watson and Crick always wanted to jump to that conclusion, and as scientists, they're much more wanting to kind of be a little more tentative and kind of figure it out first before they jump to those conclusions. And there's that bonding that we see. Nice association, they fit well together, and not only do they fit well, but it creates this nice parallel structure as we go through. One of the things that Watson and Crick, if you actually look at that article I gave for you guys, down on the bottom, if you kind of count the paragraphs, the third paragraph from the bottom, Watson and Crick even notice, it starts out, it has not escaped our notice that specific pairing would, basically what they knew right off the bat was that you could make a copy mechanism off of the structured DNA, but they didn't want to jump to that conclusion. When you're right on one thing, let's stick to that. Let's not go too much. They knew, and they even make the point, and you can read in there, further on, that might be something that we can actually look at. But for now, it's just the structure of DNA. So their discovery did lead to a possible mechanism, but they didn't actually go into that at that point. And there on is that our actual article there. One of the things I love about this article is look how short it is. It's not 17 pages. When you're right, you're right. I always remember when I was in high school or college and I had to write a paper or an essay, my best essays were the ones where I knew what I was doing. They're short, to the point, full of facts, full of information, and I felt really confident. When I didn't know what I was talking about, I would just kind of spew garbage on the paper for about 10 pages before just deciding to throw up the white flag, hoping that the teacher took mercy on my soul. So my point is this. Watson and Crick, they did an excellent job. They were able to communicate their ideas. And on top of that, not only were they able to communicate their ideas, they knew that this would be read by high schoolers, by college kids, by everybody, and it had to be able to be explained to everyone. So as you read this, and that's going to be part of your homework, notice it's not that complicated to read. You can follow along with a lot of what Watson and Crick had to say. And so what we're going to look at here coming up is how do these complementary strands of DNA actually allow for replication, okay? And those base pairing rules that we talked about earlier are going to come into play. All right. So Watson and Crick developed the semi-conservative model for this. And basically, the semi-conservative model of replication, what it does is it predicts that when a double helix replicates, each daughter molecule will have one old strand that was derived from the original. Okay, in other words, that one's conserved from the parent model, and one newly made strand. Okay, that wasn't the only idea out there. Some people actually had other beliefs. The most common ones, and I'll pause this and put them on here, were the conservative and dispersive model. And we're going to look in the next page exactly what this is. Now, Watson and Crick's data comes from this idea of the anti-parallel and the base pairings going together. You can take a look at the model. The first model is the conservative model, which basically says that the whole molecule is conserved, okay? That the whole molecule is conserved and is passed down, and a completely new template is made each time. At the end of this process, what you end up getting is 3 completely new strands, and that original strand stays intact. The second is the semi-conservative model that Watson and Crick proposed. In this case, you get the half new each time, and eventually leading to where you can see here, you do get these 2 brand new strands, but off of the parent template strand. The last model is the dispersive model, where it's parts of each parent are passed on and are intermittent, or they're interspliced with new parts. Okay, so those are the 3 basic models, and your book does a pretty good way of kind of describing each model and the experiment on about how those other 2 models were disproven and the semi-conservative was proven correctly. All right, so make sure you do look in the back about some of the experiments by Stahl and his partners about how they prove the conservative model. It's another one of those examples of using the scientific method properly and coming up with a pretty cool conclusion. The rest of class, what we're going to talk about is DNA replication. Okay, seeing exactly how DNA replicates. It is a process. There's a lot of things involved, and it should be something that you should be impressed by its speed and accuracy, and how many errors are actually occurring in the DNA replication. It's pretty small considering how massive the actual DNA is. So how do we get this process started? Replication begins at a special site, and we typically call this the point of origin or the origin of replication. And at the point of origin or origin of replication, the two DNA strands actually are going to separate, and in doing so, they're going to kind of create a bubble like this. And that bubble eventually kind of leads back together. Okay, and in eukaryotes, we can actually have multiple bubbles. We can have thousands of bubbles all at once to increase the process. Once again, it increases the rate. Why do we want to get out of this process as soon as possible? Mutations are very possible anytime that you have this going on. So make sure you get in and out of replication so you don't have things like UV coming in during this process and damaging any of the DNA. And the other thing is that speeds up the process. The replication is going to occur in both directions, going towards and away from the point of origin. Okay, and typically we call the area right here the replication fork. That's where the Y-shaped region where new DNA strands are actually being elongated. You can see this here once again in eukaryotes. You have multiple origins of replication happening all at once. Why do we have that? The purpose of this to speed up the reaction so that it happens a lot faster than if you just did it at one location. Now, when we look at DNA replication that is linear, okay, so double-stranded DNA, you're going to have many replication forks, and just to give you an idea, on a fruit fly, which is a lot simpler than us, 6,000 replication forks exist simultaneously, which increases the rate of this reaction. For us, we're going to have a lot more than that, but just in a fruit fly, you have 6,000. So the first stage is elongation of the new DNA strand. What's going to end up happening is that an enzyme, DNA polymerase, is going to play a major role in this process. And there's actually two DNA polymerases that we're going to look at, DNA polymerase 3 and DNA polymerase 1. Each one have an important role in the process. Okay. Each nucleotide that is added to the growing strand, okay, will be that nucleoside triphosphate. The rate of elongation is about 500 nucleotides per second in bacteria and 50 per second in human cells. Now, I want you to think about that. I'm going to talk about it next time, but why would you have that big of a difference? Why are bacteria so much faster than us? What are some possible reasons, both evolutionarily but also mechanically, that cause bacteria to be so much quicker than us? Along with that, some other interesting stats. The average length of the replication that occurs is about 42 million bases long in bacteria. Where in a mouse, it's only about 1,500 bases long because there are at least 25,000 areas of replication occurring at one time. Here you can kind of see the process in which we're going to need to occur. Notice the nucleoside triphosphate on the left-hand side. So this anti-parallel elongation process, the two strands are going to basically work in opposite directions, and then both sides are going to work or affect the process of it. An important thing is the direction. In DNA synthesis, nucleotides are joined one at a time, and they're always added to the 3' end, also known as the free end, of the new synthesized strand. DNA polymerase is going to be the enzyme that actually does this. And one of the reasons it is, DNA polymerase can only add nucleotides to the 3' strand. Okay? DNA polymerase cannot add to the 5' strand. So therefore, the new DNA strand always elongates in the 5 to 3 direction. So that's elongation. And elongates in the 5 to 3 direction. Okay? And it will actually read the process. It'll read the DNA strand, so DNA polymerase reads it in the 3 to 5. That's how it reads it. If it happens continuously, we're going to call that the leading strand. The other strand's the lagging. And like I said, the reason you can think about this is that replication fork opens up, that what's end up happening is the DNA polymerase, from the point of where it opens up, is going to be kind of adding the bases on one side and the other side working to kind of fill in the gaps. The side that has the gaps, that's going to be our lagging strand. And one of the terms that you need to know is that on that lagging strand, you're going to get these Okazaki fragments. And Okazaki fragments are these series of segments on the back side. And because they're incomplete sections, they need to be connected. And the connector is an enzyme called DNA ligase. It's DNA ligase. Once again, do just recall that along that template strand, the leading strand, it can only synthesize the complementary strands continuously, moving toward the replication fork. On the other side, it's going to be moving away from it, and that one is going to be the lagging. So that's the distinction between those two strands. And here we can kind of see that. Okazaki fragments here. Here's our DNA polymerase moving towards it, okay, creating one continuous strand. Backside is going to be filling in Okazaki fragments because you work until you get to this point, and then you got to stop. Another one's going to come work, fill in here, and just keep going like that, filling in on the lagging strand. Then there's your DNA ligase fixing the gap. DNA polymerase cannot initiate synthesis. It can't do it itself. So even though it's extremely important, it's not going to be the only player in the kind of the process. And one of the biggest reasons is it can only add nucleotides to that 3' end. And so what we need to have is we need to have something that comes in and does it for us. So an initial nucleotide strand is going to be called a primer. And when we look at primers, they're typically going to be an RNA primer that gets put in and laid down. And it's done by an enzyme called RNA primase. Okay, so an enzyme called primase starts the RNA from scratch. Unlike DNA polymerase, it doesn't need to have the 3' end. On the leading strand, you just put down one track of primer, and then from that point on, it just goes. On the lagging strand, each Okazaki fragment needs its own individual primer. So there's a lot more gaps on the lagging strand of DNA. And then I'm basically going to show you kind of a short animation of this by working through. So the first thing, here's that primer being laid down. In this case, we're laying down an RNA primase. From that, we're going to get the DNA polymerase to come in and start laying down its bases, following the base pairing rules. A goes with T, C goes with G. Then, eventually what's going to happen is it creates this little small segment. You see another primer had to be laid down. Another DNA polymerase is going to come in and fill it up all the way till you get to the primer. It stops. There's this gap right here of primer, and that's going to have to be removed, okay? And what's going to do that is another enzyme similar to DNA polymerase 3, DNA polymerase 1 will end up replacing the RNA with DNA and getting everything except for this little gap here. So DNA polymerase 1 is the one that's going to come in and remove this primer. DNA polymerase 3 is the one that laid down the original track. Last guy, ligase, seals up the track, and now we have the complete lagging strand. Here's all the enzymes that you need to know. I'm telling you, on our next test, which will be over this chapter and the next one, you will need to know all the different enzymes. I will give you a diagram very similar to this, and you're going to need to label DNA polymerase, okay, primase, excuse me, the RNA primer, ligase, DNA polymerase 1, DNA polymerase 3, the helicase, and then a couple other ones that we're going to talk about now, like helicase, SSBPs, and topoisomerase. I will also expect you to label the lagging strand, the leading strand, which is a 5, which ends a 3, all those types of things. So what are the other ones that are important in this process that we're going to kind of go over that you will need to know? The first one is helicase. And helicase, its job is to untwist the double helix and separate the template DNA strand at the replication fork. The next one is the single-stranded binding proteins, also known as SSBPs, and that's fine if you ever say that in class or put them on a packet. The single-stranded binding protein's job is as the DNA opens up, it needs to hold it open. It's going to want to seal right back together, and so when you get that replication fork, they'll kind of form up here, keeping the molecule open. Topoisomerase's job is to work up ahead of the replication fork. So it's going to work way up here, As this opens up, it's going to kind of create a kinking that occurs, and topoisomerase's job is to overcorrect and fix the overwinding that's up ahead. The specific protein that does this is the enzyme called DNA gyrase, which is Gyrase. It's a classification of topoisomerase. And like I said, its main function is to prevent the supercoiling. Those are all different examples of some of the proteins that you need to know. Other ones, we talked about this already, but just as a summary, primase, its job is to lay down primer to get the DNA replication started. And what it does is it allows for a 3' OH group that allows DNA polymerase 3 to start laying down its bases. Okay, because RNA polymerase does not, or excuse me, DNA polymerase cannot do it only on that 3' end. So it's really important to get it started. DNA polymerase is the one that's going to lay down the base pairs. DNA polymerase 1 removes primer and then also does the fixing and filling in the gap. Then finally DNA ligase sews up the Okazaki fragments. And this diagram is from your book. Once again, if I was a betting man, I think I might take a diagram that looks identical to this, cross these words out, and just put numbers and have you answer all of this stuff. So I would memorize this. Not a bad idea to draw this a couple times. I remember when I studied for this stuff, it worked for me when I actually saw it replicated a couple times. Okay. All right. The last thing I'm going to talk about this kind of briefly, just realize, especially initially we didn't think this in science, but we now know that the whole thing is a machine that's kind of linked together, and it's probably not stationary in the process. So a lot of times people think these, and I even seen animations and used animations in class where they all kind of come in individually. That's pretty much not the case. We now think that the molecules more likely come in as a big machine, and it's that the machine is actually is stationary while the DNA actually moves through it. And here you can kind of see that more realistically represented with the shape of the molecule, all the enzymes kind of connected here as one big unit, and the DNA being dragged through the entire process. What we're going to look at next is the proofreading and repairs of this process, and that'll kind of wrap up this section of notes. DNA polymerase does have a mechanism to backtrack and be able to proofread and read materials and fix any mistakes. Okay, and one of its abilities is to see if there's any issues. One of the most common problems that we can have is what we call a thymine dimer, and that's when you have two T's that are bonded together instead of bonding with the A's, and because of that, that's a pretty big mistake. So what ends up happening is DNA polymerase can come in and break that bond and get them back to where they originally should be. And here you can kind of see that thymine dimer being fixed by DNA polymerase. And once again, it allows to read, backtrack, proofread, fix types of mistakes that there may be. One of the problems with this is that there's no way to complete the 5' end. So what ends up happening is the DNA molecule is going to get progressively shorter over time, because you remember on that with the lagging strand, you need to lay down that primer, and therefore there's no way to kind of stop here. Let me show you that. So what ends up happening is that there's this area right here that ends up being shorter. We're losing information that is not being coded. Now what's really cool, and this is one of those awesome evolutionary adaptations that we have, is that we're going to have something on the end of our DNA which basically is going to be useless, that we can lose and not affect and not have to worry about it being not replicated. We call that a telomere. So the telomeres are found at the end of the chromosomes, at the end of the DNA, and once again on both sides. And what it does is, if those parts are lost to erosion of the gene, it's not going to affect it. So the telomeres are like useless DNA that we don't need, and over time those are slowly going to be eroded, but it's not going to affect genes that we actually use. Very cool adaptation. And here you can see these are the telomeres at the very ends of the chromosomes there. All right, that's all I got kind of for the notes. You did get that packet about nature, the nature article. Here's what I'm going to expect you to do with that. So I want you to read the article. Read the nature one, read the one about DNA replication, and read the discovery of a new class of damaged proteins, all three of those. And I want you to think about this question. I want you to think about how are those related and how are they related to what we're covering in class. So just type a one-page summary of the articles, how they're related to each other, and how they're related to what we're talking about in class. And we're going to turn that in next time. Okay, so read them, summarize, how are they related, and how are they related to what we're talking about in class. That's all I got. Hopefully there's going to be a snow day tomorrow, and I'll see you guys on Monday.